actually addresses a question related to early organogenesis, and different signaling pathways involved in this. There are different morphogens which are involved in these processes, and there is a lot of information available about all these processes. Now, we all know when it comes to development after fertilization, there are different lineages are formed, and these lineages eventually gives rise to different organs. And as I mentioned, there are different signaling pathways which are involved in this. A lot of morphogen molecules also are involved in giving specific identity to each of these lineages, which eventually leads to formation of the specific organ. Now, my interest lies at this stage where the lineages are formed, and once these lineages forms organs, how these organs are maintained, because in the adult stage, these organs undergo various kinds of injuries, and now there are various reports of adult stem cells which shows that there is resident pool of stem cells which essentially divide and replenish the damaged tissue. Now, to understand this question, as Vijay mentioned, we use a simple system which is flight muscles, and when we are addressing this question, it led to discovery of a stem cell. Now, these are the stem cells, and the population in the green are the myoblasts. And what this thesis says that only cells which are dividing in this whole population is stem cell and not the green cells, which is the classical view. So we refute this classical view and say the only cells which are dividing are stem cells, as shown here. And this was all done by using simple genetic tricks and cell counting. Now, we all know at least from Vijay's lab, the flight muscles are of different size, and they have different functions, different neuronal connections, and they essentially arise from this pool of cells which are known as myoblasts. Now, Nagaraju's work and Priyankana's work and many others from the lab have already shown that these cells during metamorphosis fuse to form these different kinds of muscles. Now, it's remarkable that different kinds of muscles having different function essentially come from this handful of precursors which are in the thoracic segment 2. And my job was to look at how these cells really divide during these larval stages. Now, as we can see, there are only 10 cells as this study shows, and these 10 cells divide during different stages to form 2,400 plus number of cells. We followed this cell number by actual counting, and what we observed that this was the stage when the counting wasn't really going well. So there was anomaly in terms of the number of cells in the earlier stages and the number of cells in the later stages, which I'll come to in a while. Now, the question which is central to this thesis is, if there are few precursors, namely adult muscle precursors, how they divide in during different larval stages to form these thousands of myoblasts, which eventually form muscles of different function. Now, here is a 3D reconstruction of 3D reconstruction of a wing imaginal disc, and essentially myoblasts are found on this tissue which is known as epithelium. Epithelium is an ectodermal origin, myoblasts are of mesodermal origin, and throughout their larval history these myoblasts are tightly stuck onto this epithelium, and as we can see, they can be marked by using a twist antibody. Now, this arrangement is essentially similar to a rice in spoon arrangement, where rice represents myoblasts and the spoon represents epithelium. And we can appreciate there are different layers of these myoblasts when it comes to third insta. Besides that, these myoblasts show great variation in terms of their size. The myoblast layer which is next to epithelium in close contact are the smallest in size, and this was something which was new in these kind of studies. And the cells which are at the farther distance from the epithelium, they are really large in their size. Now, to understand how these cells really divide, we started with 3 simple hypotheses, saying if adult muscle precursor divides, it can form 2 cells which are of equal potential. Now, these 2 cells will divide to form 4 cells, and so on. So that's a symmetric mode of division. The other hypothesis was if the cell actually behaves like a stem cell, where only one of the daughter at the end of this first division continues to divide from a stem cell and a differentiated progeny. then we'll get a asymmetrical growth which will be linear. So in this case it will be exponential, in this case it will be linear. And for argument's sake, we thought there will be another mode of division where the cells employ symmetric in the start, amplifying stem cell population, and then switching to asymmetric to form differentiated progenitor, which will eventually form different kinds of muscles. To understand this particular and validate hypothesis, we use a simple genetic trick of cell labeling. It's popularly known as twin spot Markham. And the way it works is whenever there is a cell which is actively dividing, you can label the daughters with two different colors. In this case, GFP and RFP. And once labeled with this GFP color, this cell continues to express GFP. And all the cells coming from this lineage are labeled green in color. Similarly for the RFP. And so one can monitor after the first division, when the daughter cells are labeled, how many times these daughters have divided in given period of time. And if they divide same number of time, one should get equal number of green and equal number of red cells. But if this cell gives rise to a stem cell like cell, and if that stem cell gets a green color, and if it divides more number of times, one would get more number of green cells. And if the other cell shown here in red, if it doesn't divide and takes post-mitotic phase, then you will see only one single red cell. Now this was the, this is what twin spot Markham allows. Different papers have used this technique, and there was a very good study from O'Brien et al. Where they showed based on nutrition signaling, under starvation cells choose to undergo asymmetric division, and this method was employed and it showed very clear-cut results. Now the protocol for this is simple. It starts with a heat shock which induces expression of flippase, and this flippase indeed combines DNA where the cell labeling takes place. Now we have done lineage trace from the early instars where the heat shock was given in the early instar and the clones were retrieved in the late instar. Now during this stage, which is essentially larval period, the cells which are labeled are dividing. And at the end, when we cut open this larva and look at what are the colors and what are the sizes, one can say evidently that if the clone size are same, the cells divided symmetrically. If the clonal size is different, then one has to think about stem cell hypothesis. Now this is the first clone which was triggered in the early instars and recovered in the late instars. Now they have divided so many times, and what we can observe that there are equal number of green and equal number of red cells, saying retrospectively when the clonal analysis was done, which was first instar, cell divided to form cells with equipotent potential in terms of division, and that's why we got the same number of green and red. Now remarkably, when we were doing this experiment, in the late instars, cells show different pattern of division. As we can see, there is larger green clone where there are 6 cells and there is one huge red cell. Now this red cell has a single nucleus, other cells also have single nucleus, but there are 6 green cells and only 1 red cell. This showed the cells at this stage, which is late second instar onwards, were dividing asymmetrically. In summary, whenever we perform clone analysis in the early instars, we always got symmetric clones, saying it was symmetrically dividing at that point. But late second instar onward, the clones were asymmetric, and always there was a one large clone having many cells and one small clone which had only one cell. In summary, we discarded hypothesis number 1, which was symmetric division, hypothesis number 2, which was asymmetric division, and say the cells actually employ both symmetric division in the early instar, which is first instar and the second instar. At the end of second instar, the cells start dividing asymmetrically, forming a post-mitotic myoblast and a stem cell. Now, this was very interesting because this was not observed in any cases, but it was happening at a very particular stage of development, which was late second instar onwards. So we started looking at what was happening anatomically in this tissue around these stages. As we can see, the myoblasts are labeled here in the green, and the epithelium onto which they are sitting here is in white. And it can be seen very easily that the cells in the early instars are monolayered. And this monolayer arrangement eventually changes to multilayered, and this was also not reported in previous studies. So we were wondering what exactly is happening in these stages. Now to see how many cells are actually dividing in the late insta, because this was the time when the cells were dividing asymmetrically in stem cell fashion, we use phosphohistone antibody. Now this antibody allows labeling of mitotically active cells. And what we could see, out of these thousands of myoblasts, there are only few cells which are actively dividing, and in this case there were only 40 cells in the third insta. Now this said that there are only 40 cells, and possibly these are the only cells which are dividing and giving rise to myoblasts. So, and other important observation we made was cells were dividing only next to this particular tissue which I told you guys, epithelium. Now at this point we were wondering, if the cells are dividing and they are dividing next to epithelium, there might be a possible signaling coming from epithelium, signaling these cells to divide. And as in when these cells move away from the epithelium, they take post-mitotic fate because there is no more growth signal. Now we validated phosphohistone data by using EDU labeling. EDU is thymidine analog. It gets inside DNA when it's replicating, and it can be probed by using click chemistry. So when given with a pulse of EDU, label was observed only in the layer next to the epithelium. But when the pulse was followed by a chase of couple of days, the label got diluted in the first layer, saying the cells divided. But the label was seen clearly in the third layer, which is the farthest most layer, and this showed that if the intensity of the label in the third layer is maximum, this cell never divided and took the post-mitotic fate. Now, in addition to change in layered arrangement, we thought how the cells might be dividing in the different layers when it comes to early developmental stages and the late developmental stages. To address that, we use this particular construct CNN-GFP. Now CNN marks centrosome, which enters inside centrioles, and whenever there is mitotic division, one can see centrioles position at two poles and the DNA in between them. Now based on position of centrioles and mitotically labeled DNA, we saw that in the early insta the cells divide along the epithelium, and this is the stage when the cells were dividing symmetrically. Similar experiments in the later stages when the cells were dividing more like stem cells, what we observed, the division pattern was different, and now the division axis was shifted to more of orthogonal, such that the cell which was in the second layer loses contact with the epithelium, ensuring its post-mitotic fate. So in summary, the only time the cell divides is when it is in contact with the epithelium, and the cell gives rise to a post-mitotic. When the cell moves into different layers, it actually undergoes changes in terms of cell size. So what one can see here is the cells which are dividing actively, they are very small. So we call these cells as stem cells because these are the cells which are actually dividing. The stem cell eventually gives rise to cells of different dimensions, and this is the cell which shows dimensions very similar to the red cell which are shown in the first clone, saying whenever a cell takes post-mitotic fate, it shows characteristics of differentiation. Now this summarizes wild type characterization of how cell divides, and in summary they divide symmetrically and asymmetrically later on. Now this brings us to what are the possible molecular mechanisms which govern this whole division. And considering there is great knowledge available when it comes to asymmetric division from the neuroblast literature, we thought the similar molecules might be important when it comes to asymmetry in the myoblast division. So we propose it might be the NUM and Notch, which are known molecules when it comes to asymmetry, and here is Notch pathway. Notch is a membrane-bound transition factor. The only time it activates Notch pathway is when the ligand binds to it, which leads to cleavage of this transition factor entering inside nucleus, and that's how the Notch pathway activates. So it needs a ligand when it comes to activation. Now NUM, on the contrary, is a cytoplasmic molecule, and NUM downregulates Notch pathway. So it binds to this Notch intracellular domain and triggers its degradation. So if there is high Notch, there is probability of high division, and if there is NUM in the cell will not divide because of inhibition of Notch pathway. To address this, we started with immunohistochemistry of this tissue, and as we can see, wherever there is high NUM, this cell shows very less notch staining. Now notch is there in all the cells as can be seen in the GFP marking all the myoblasts. Now this says if the notch is there in the cells and the numb is in the few cells, it might be that it is differentially regulating different cells. To understand whether the numb essentially controls myoblast division or not, we use RNAi specific to numb protein, RNAi against numb gene. And whenever we downregulated numb, as can be seen, phosphohistone count went up in comparison to control, saying numb is important when it comes to myoblast proliferation. Now similarly, when we did notch RNAi, it led to decrease in the myoblast proliferation, saying numb and notch molecules are important when it comes to division. But when we upregulated notch by using notch intracellular protein, what we observed, the myoblast proliferation actually doubled, saying numb and notch are important when it comes to myoblast proliferation. But the real taste of their role in asymmetric division was something where we would subject these cells in a time period when they were dividing asymmetrically, but now the conditions are different. These cells, when they are dividing, we take down notch pathway by activating it and giving both the daughter cells equal advantage of division. And in this case, when the analysis was done, it led to symmetric spots of GFP and RFP. Similarly, when we downregulated numb in the late third instar and triggered clonal analysis, it led to symmetric spots. Now this essentially says if the cells are dividing asymmetrically and you take down the molecules of asymmetry, it will revert back to symmetric mode of amplification. Now one can appreciate the clones actually shows very different distribution than the clones we observed in the start. These clones are all over the epithelium, they migrate long distances, which was not observed for the wild type clone analysis, saying indeed whenever you downregulate these molecules, it leads to sort of metastasis in this particular population. Now the next question was, if the cells are dividing only when it comes to their contact with the epithelium and they do not divide when they reach further layers, was there any sort of signal coming from the epithelium? Now when it comes to Drosophila, there are only two ligands when it comes to notch pathway, the delta and serrate. We probe for both, and what we observed, serrate as shown here in red, specifically expressed in the epithelium, and it can be seen that this makes very close contact with the actively dividing cells. So to test this, whether serrate is actually activating notch or not, we downregulated serrate by using epithelium specific GAL4, and it led to decrease in the myoblast proliferation as shown here. It also led to decrease in total myoblast count as in comparison control, saying serrate comes from the epithelium, it activates notch which is in the cells which are right next to the epithelium and gives rise to a differentiated progeny and a stem cell which maintains contact with the epithelium and continues to divide. Now with that I summarize this first part. When it comes to mitotic division, these are the stem cells, they have high notch in them, and the cells which do not divide, they are post-mitotic. Now having explained this, it doesn't explain what actually regulates numb expression and when it is actually regulated by which molecule. What we observed, yes?
So in your notch upregulation,
Yeah.
you see increased proliferation.
Yeah.
Did the number of cell layers increase?
Yes, actually it increased. I skipped that slide. I can show you that. Yes. So whenever we actually upregulated notch, activated notch, it led to upregulation of in general proliferation and number of layers. Yes. Yeah.
But you still continue to observe the pH3 only in the layer adjoining the epidermal.
No. So when we did phosphohistone analysis in this background, interestingly, phosphohistone was present in all the layers, and also the division axis which we observed was not something which was there in the wild type. So whenever there is a division in the third insta, the cells divide in such a way that the differentiated progeny enters in the second layer. In this case, the spindle orientation was more random, and we could see division in all possible directions. And that's why when you look at this particular clonal analysis, you see cells going all over the wing disc. Yeah. So now the issue with the numb notch signaling was it was only seen after late second insult onwards, and so we thought what can be the molecule which possibly regulates this. So wingless was known to express in the epithelium in the late second insult onwards. We thought this might be the molecule, and in short what wingless does is whenever there is a wingless ligand binding to frizzle, it leads to stabilization of beta-catenin. Now beta-catenin is a nuclear effector when it comes to wingless canonical pathway, and after that it activates wingless specific genes, and in many cases wingless regulates proliferation at the same time. Now here we are trying to propose wingless regulates NUM, and to test that we started with immunohistochemistry, immunostaining, showing that wingless essentially comes in the notum epithelium in the center. Now what one can see, essentially this expression of wingless is in very close contact with the myoblast layers, and so thus it possibly pointed towards that wingless, considering it's known to be secreted at longer distance and affect larger population of cells, we thought wingless might be secreted by these cells and might reach all the myoblasts and thus pattern them for the asymmetric division. To test that, we did a beta-catenin staining, and what we observed in control, the beta-catenin activation is there in all the myoblast layers as shown here. But when we used a mutant for wingless pathway, which is specifically downregulates wingless expression only in the notum epithelium, the NUM expression was, the beta-catenin activation was down. It also resulted in less number of myoblasts, saying wingless regulates proliferation of myoblasts when it comes to third insult onwards. Now experiments on NUM line showed that whenever we use the same mutant which takes down wingless pathway only in the epithelium, notum epithelium, it showed a disappearance of NUM, saying wingless along with regulating myoblast proliferation regulates NUM expression, saying when it comes to asymmetry, it's a wingless induced NUM which breaks the symmetric division, makes the cells undergo asymmetric division, and this happens characteristically after third insult onward. Now we went one step ahead and downregulated both pathways in these proliferating cells, and what we observed, whenever we downregulated both pathways, it further reduced the number of layers, but interestingly it also showed a phenotype in the cells which were harboring the epithelium, which were harboring the myoblasts, and this was something remarkable. Now this shows that when it comes to myoblast proliferation, if there are less number of myoblasts, it also leads to wrong patterning of this other tissue marked here in the blue, and this tissue eventually forms a attachment site for the muscles, saying possibly the stem cells which were in contact with this epithelium are signaling back to the niche. With that we come to this part where I say there are few precursors dividing in different larval stages. In the early stages they divide symmetrically and the division axis is parallel to the epithelium, and in this case it's a serrate notch pathway which is important. Later on serrate notch signaling switches to serrate notch plus Wnt, which induces NUM expression in the cells which are post-mitotic, and this leads to asymmetric division. So in summary, wing epithelium acts as a niche, there are stem cells next to them, and there is heterogeneity in the total myoblast population which is different than the classical view. Now having said that, this was all happening in the larval instarts, but in the development people have adultocentric view where all the development is essentially for this particular fly which comes out of the pupa, and the preparation is happening in embryonic stages and in the larval stages. So the new question in front of us was if there are stem cells and if there are post-mitotic cells. And if the post-mitotic cells are the cells which form muscle, what happens to the stem cells in the adult fully formed muscles? In other words, are these cells of any significance when it comes to adult musculature? Do these cells stay back unfused in the adult fiber? To understand this, we devised a simple experiment where if the cell fuses, the GFP will be lost. Now, for example, if this is a myoblast stem cell coming from the population, and if it has the GFP on the membrane, and rather than staying unfused, if it fuses to form muscle and that's its nucleus, the GFP will be lost on the muscle membrane. But if it doesn't fuse, as shown here, one should be able to see a single cell which has a GFP all over it and single nucleus inside it. Now, to address this particular question, we did a clonal analysis marking actively dividing cells, stem cells in the early, late third instar during larval stages, and we chased these cells in the adult fiber. So this is adult fiber. The blue showed all the nuclei, and the green essentially are the unfused cells which have intact membrane around them. Now this was something very interesting which was not seen any time before, and when looked for their position in the fully formed muscle, which is labeled here in red by using phalloidin, what we can see, the cells occupy niches very similar to what is known in case of vertebrates. Now this is very interesting, and considering flight muscles which demand extreme performance when it comes to flight and in general life of flying insects, we thought these cells might be very similar to what is known in case of vertebrates. So there are specialized stem cells when it comes to adult muscles. They are known as satellite cells, and we propose here that these are the cells which are very similar to satellite cells known in case of vertebrates. We confirm our finding of unfused cells next to fiber by showing that there is one cell which has an intact membrane, a single nucleus, which is crescent shape, very similar to what is known in case of vertebrates, and it is right next to the muscle fiber as shown here by a membrane, and it is surrounded by ECM matrix. It shows great differences when it comes to its cell size in general, and the nucleus characteristically is very small when compared to the nucleus of the fused nucleus of the muscle. Now we have done many experiments to show that upon injury by using different means, as simple as sticking a small dissection pin inside fly thorax, that these cells upon injury proliferate and eventually fuse to form fiber and recovers the damage. We have essentially repeated all the experiments in different Drosophila species and also in honeybees, saying this phenomenon is essentially conserved when it comes to evolution, and similar signaling pathways are important when it comes to proliferation. So here in this case I have done very similar experiments where I take a honeybee of 7-day-old Apis florea, and I give an injury by using a dissection pin, and I look for possible expression of phosphohistone which marks actively dividing cells. The only time one sees those cells is when the cell proliferates, and this is only in the injured background. So in summary, what this thesis says that when it comes to adult muscle precursors from the early embryonic stages, they divide symmetrically in the start, asymmetrically later to form myoblast pool. There are 10 in number when it comes to embryonic stages, which divides to form 2,500 number of myoblasts, and there are around 250 stem cells population which are in close proximity of the epithelium. Some of these stem cells remain unfused in the adult fiber, possibly required for the regeneration, and most of the post-mitotic cells eventually form the muscles. Now this leaves us with many open questions in the field, that if there is any regeneration happening, is it happening in all the muscles, and if it is happening, what are the rejuvenation factors required for continued maintenance of this stem cell pool. Now we have just shown serrate and wingless to be important when it comes to niche signaling. There might be more of those signaling required for stem cell maintenance. We have not found a stem cell marker in the embryo larval stages, so that stem cell marker is something which is important to show that these are essentially the same cells which stay back in throughout the stages. And interestingly, this thesis also raises question of whether stem cells signal back to niche for its own maintenance or not. And obviously, studies till now have not shown any parallels when it comes to vertebrate myogenesis of symmetric and then switch to asymmetric division, because what we think, it might be the common strategy employed by vertebrate muscles and in general any organ, because there are increasing reports of stem cells in the adult tissue. So it might be that there are always pools of these stem cells which essentially forms the muscles. So with that, I'll take questions if there are any. True, true. So whenever a cell divides, if I understood your question, you need num in addition to being these cells being away from the epithelium. Now whenever a cell divides, it has a residual activity of notch. Now that can actually cause proliferation in these cells. Having said that, notch actually even in smaller amounts can bring all those changes. Now that's the first part when it comes to num notch signaling. But essentially asymmetric division, which we are looking at, is important when it comes to giving fate to any cells. And I think num classically, though it is known for downregulating notch pathway, the actual action of num in other respect, whether it regulates other genes or other proteins which bring down, brings differentiation or not, is not known. So I think, besides function in regulating notch, I think it might have other functions as well.
Expression, did you monitor in the adult stem cells whether it has an impact on the muscles in the adult cells?
So notch is known to overexpress twist, and twist indeed causes overexpression of notch. Now twist marks cells which are in a naive state. If I overexpress notch, it thus leads to extra proliferation, and it's known by Shumita from the lab, Shumita studies from the lab which are published long back, that if there is high level of notch, these cells never take myogenic fate. So these cells continues to divide and never commit to a myogenic fate. So muscles are not formed in this particular background. But if done in stage specific way, let's say you do overexpression of notch only in the adult stage, then there is huge proliferation. And again in this case, what's known is notch should be followed by activation of Wnt pathway. And this studies comes from lab of Thomas Rando, and essentially it's commitment to the myogenic fate which is brought about by the Wingless pathway, and that's what we see here. So yeah.
In downregulation of num, you have over proliferation.
Yes.
And in the second and third layer you have notch expression. So how do the ligands react with the notch to give the proliferation?
Yes. So as I was telling Anup, actually whenever a cell divides and if it was undergoing notch mediated activation for proliferation, this cell has residual levels of notch in it. Now the inhibition is not there and this cell divides. Now will this cell continue to divide is the question, because it is not getting any more signal for the division. Now this, what we observe in phosphohistone analysis, the only time you see phosphohistone even in num was only in the second layer, never in the third layer. And also the division axis was something which was very interesting. It showed division axis which was essentially parallel to the epithelium in the second layer. These cells never divided even when they had no num in them in the third layer on. Yeah. Yes.
So as the cells start dividing and come away from the epithelium, you show the cell size also increases, and I assume their properties are also different. So what could be the underlying significance? And the second question is the change in the cell size, Is it because of its loss of contact of the signals coming from epithelium?
Yes. So what's known in case of cell size and their contribution to muscle development is, in case of DVM formation, in case of DVM formation, which is another group of muscle which is found in the fly thorax, there are these cells which have essentially very long extensions, and the job of these cells, and I think they are doing the same job here, is to go and form the first set of fusion myoblasts. Considering they have longer extensions, they go and essentially attach muscles and start fusion process. Now that's one hypothesis which I think. The second hypothesis, and there is data for it, is from Tom Kornberg's lab, and what it shows, these cellular extensions are used in transfer of signal from the epithelium to the myoblast, and these myoblasts which have longer extensions, and what he calls them as cytonemes, which are thin hair-like projections, are used in passing this signal to another tissue which is trachea. So if you take down these cytonemes by using genetic means, it leads to less proliferation in tracheal zone, and thus not well formed trachea. So that's one. Second is there any significance, why the cells are showing different sizes, and is it because whether they are losing contact with the epithelium or not? Now what we know from the lab study and Sudarshan's paper is wingless induces vestigial, and vestigial is known for development of the flight muscles. Now what we think here is vestigial and the downstream pathway molecules of wingless are causing these cells to take post-mitotic phase. And when it comes to post-mitotic phase, they are activating different genes required for different fibers. And we think that they are pumping in a lot of mRNAs required for those muscles. So, and essentially the cell size denotes that they have activation of many genes.
Yeah, so you have looked at in the third instar phase, wind is one of the major factor that leads to this asymmetricity. So have you looked at in the earlier phase, wind is at all not present, or is it being expressed only in that stage? And how is it?
That's a good question. So it's known from, yes, and not included slides for it. So I'll take you to the backup slides. I have very interesting data on that. So when we were looking at wind story, I did do the profiling. I looked at epithelium in the early instar and late instars, and wingless actually comes only in the third instar, and that was something which is very important. Now what if you do other way around? What if you put wingless in the early instar itself? What will happen? Now when we were doing this wingless story and seeing that indeed it regulates number expression, there was a paper by John Paul Vincent's group, and this is what the paper says. If wingless is a morphogen which acts over a long distance, it has to be secreted from a cell which is produced and reach longer distances. He says it's not required. Even when the cell producing it do not secrete it, it will lead to the same patterning. Now in our tissue what we see, these are the cells which are producing, and the myoblasts are essentially here. And if he was correct, then we were wrong, essentially. And so the way the experiments were done, I took this wingless, NRT wingless flies, and I looked for whether they show similar levels of beta-catenin activation, number of myoblasts, and in general. And what we observed, this is a remarkable result. This is a very rare result in cherry picking of data, let me tell you upfront. And what we observed, that wing disc, which essentially has only one pouch and a niche which is notum, was not seen in this particular cell, organism. And what it gives in terms of glimpse of niche importance is, whenever there was a duplicated pouch, there was no myoblast. So in short, if there is high wingless in the start, it will lead to differentiation of the stem cells and less number of myoblasts.
Rajesh, you showed that in the third instar, there is division only in the myoblasts that are immediately next to the epithelium.
Yeah.
So what would you expect would happen if you killed the epithelial cells with something like HIT?
Yeah. So this experiment essentially says that if the niche is absent, these cells will not proliferate. Now, having said that, it's known in many contexts, if you remove niche, these myoblasts, these stem cells either die or they migrate to different niche. Now, I'll take this question in different direction. Ishwar once mentioned studies from the Nipam's lab, and what he said that whenever in the early development, whenever he removes germline stem cells, which also comes from mesodermal origin, removal of germline stem cells leads to, he observed that whenever you remove germline stem cells, they come back and you see the normal germline development. Now, these experiments were done in the very early development, and considering both myoblasts and germline stem cells come from mesoderm, I think what he observed was mesodermal cells replenish the germline stem cells. And what I think, if I remove, in this case, niche by using HIT or something, these cells possibly will migrate to another niche or they will undergo possible death. Yeah.
Wingless in the hinge region, and you also have it in the DV boundary. So
Yes.
do you see any signs of the myoblasts in the double pouch experiment going to the wrong place?
So considering this represents a snapshot of what was happening back in the development time, but what is known, myoblasts for the flight muscles and myoblasts for the mesothoracic leg come from the same precursors, and that is decided very early on in the development. So possibly these cells have migrated to some other location.
But not.
Problem with this experiment is you're altering the wingless pattern of expression in the entire animal
Yes.
early in development. So you had a duplicated pouch.
Yeah.
So the question is, if instead of doing that, you temporarily alter it so that after the wing has been patterned, you remove it from the presumptive notum.
Yes.
So that you have the tethered wingless only in the presumptive notum, then what happens? If your point is that wingless is acting as a gradient there,
Yeah.
then you would have only myoblast stem cells proximal to the tethered wingless proliferating as stem cells, and the other ones proliferating asymmetrically, and the other ones proliferating symmetrically.
Yes.
That experiment
Yes.
is not easy to do. But
Yes. Yes. Actually.
so this experiment is really, in some sense, is developmentally too early to answer that question.
Right, but I'm curious to know, even if it's theoretical, if you remove wingless from the notum, the patch of wingless expressing cells, are myoblasts at some point attracted to the wingless in the hinge region and the DV boundary? Means, are things being secreted from there too, or is it too far away?
So what I understand from your question is, if the wingless is removed, will the myoblasts still be in the notum region, or the wingless actually brings these myoblasts in the notum or not? Yes. 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 So removal of wingless actually in this case do not lead to migration of myoblasts, because myoblasts are there from very early stages, and what we understand is in the first instar the myoblasts are all over the wingless. It's only in the second, the end of first instar onward. In the second instar, they are found in the notum region. Okay, and this is the same time when the notum is defined. Now, we think that there are other signaling than wingless which are attracting them to the notum. Okay, and the reason I'm saying this, because in this particular case, the notum was altered to become pouch very early during development, because this essentially says whenever you downregulate, whenever there is upregulation of wingless, this part of the epithelium leads to pouch formation. And considering it has happened in the early instars, we think the myoblasts have not migrated or they have died.
So side point related to the Alexander et al. Paper is they looked at third instar wingless and found that the subset which actually has a strong effect on pattern.
Yes. Yeah. Yeah. So there are two people distinctly. Yeah.
So this experiment really, unless it's done in the third instar larva,
True.
doesn't
Yeah.
have
Does it?
very
Yes.
strong
Yes.
myoblasts patterning that
Yes.
goes around with
Yes. True.
Okay, I can.
Yeah.
So you use the term niche to
Yes.
to define the location where the myoblasts are sitting.
Yeah.
Very intimately in contact with the epithelium. And in developmental biology, anyway, traditionally there is signaling between different germs and layers. So this question has two parts. One is, why do you use the term niche over here when it's just two layers signaling to each other? And the second is, you also show that some of these stem cells survive in the adult.
Yeah.
So where is the niche over there? How is the
Yes.
transition taking place?
Yes. So the only reason why we are saying, I mean, the epithelium as a niche is because the cells which are right next to them, they are dividing, in a stem cell fashion. Now, going by the definition, whatever signals, stem cell to actively divide and orchestrate and, plan their further division is niche, and that's why we chose niche. Now, there are many examples of that, and also in case of intestinal, intestine development, where essentially there is, layer of muscles which signals these cells from the intestine to undergo asymmetric division and so, and that's why we chose. Now, the other thing which we say in this particular study is this is a transient niche. So it's not a niche which is seen in normal context. It's a transient niche. It's undergoing changes from every stage. In the early insights, it's serrate, in later insights, wingless. And what we know from different stages, different studies, is there are a lot of molecules which pattern this, all the epithelium in the pouch, in the notum. And there are, these signaling molecules, and we have other results saying all these signaling molecules also pattern this tissue. So based on that, considering these cells eventually gives rise to different kinds of muscles, and it essentially we think is resultant of these cells which can form post-mitotic cells, essentially of different identity, and we consider them as a stem cell, and the cells which are signaling eventually become niche. So we had reviewers comment on that when you say as transient niche, you should refer to a particular paper, and that paper also used the same term. So it's actually borrowed from previous literature which is known in case of some other organ development. Now in the adult, what we know till now is there is debate going about niche, where exactly these cells are, and it seems in first go the stem cells are located all over the muscle fiber. Now having said that, I have been in discussion with many people in the conferences and nobody knows about them. Now what everybody is proposing is there might be some signals which are coming from either neuronal connections, and these neuronal connections might be the places where majority of the stem cells might be residing, but there are no studies. So even in our study we see they are present in all this locations when it comes to muscle fiber. So he's my junior, he knows my study. So we can monitor it. I didn't tell him that question, Vijay. So we did actually, and it's easy because there is this technique, clonal analysis, and we have GAL-force, very specific to this tissue. Now what it allows, along with Markham, is whenever cell is dividing, it gets its GFP based on a recombination during mitotic phase, and that's how we can see whether the cells are dividing or not, or the cells are of mesodermal origin or yeah. So at this point we don't know whether it's a symmetric or asymmetric, but this is the hypothesis again, that if somebody has to call a cell as a stem cell, it has to undergo a division where it self renews and gives rise to post-mitotic progeny. And till now what it looks like, there are symmetric divisions happening, and what is known from vertebrate literature is whenever cell divides symmetrically in this particular context, it divides parallel to the fiber. So if this is the fiber, the division axis is essentially parallel to fiber, and this studies comes from the lab of Michael Rudnicki, who found the molecule which identifies the stem cells in the adult fiber. But this field is full of controversial studies, so and this is one of those where they show in the adult where notch and numb is important, and then others try reproducing it. In some cases you see those results, some cases you don't. If there are no more questions, I have very small acknowledgement slide. Now, as I told you guys, my start was bumpy, and I was not listening to Yamuna, and there were different reasons for it. And I'm happy that she knew about that. But then I was exceptionally very lucky to be in this group, and so a lot of times when you are on campus, you want to go home. I never felt like that, and my folks at home are very difficult people to deal with. So to be very frank, and my mom is, the best critic I have found in my career. And I remember actually when I found the first time that upon injury the cells divide in the adult, she said, it must be there. It's known. And I was like, I think I'm talking to the wrong person. And I went back, I looked at PubMed, and I was like, no, this is the first time. So, they supported me a lot, and I lied to them, I'm working hard, but I'm in canteen. So, these are fantastic people, and they helped me a lot, and always whenever I needed any help, because when I changed, I didn't know anything about Drosophila. And a lot of these people are not there, but they helped me a lot. There are a few new additions, such as Dhananjay, who's working on myoblasts project, and Shankar and Bhaskar here. I have cropped him such that he is not visible so much. Yeah. So, and then there are a lot of people, who have gone for their postdoc, but I'm still in contact with them and exchange my ideas. And these are the best people I have found, and I think the credit to what we have seen till now goes to these people. More importantly, I have discussed my science and taken a lot of time of these people is, obviously Vijay and Heinrich. So all the credit of what we have seen goes to these two people, and I think one would miss a finding like this if you don't have out-of-box hypothesis. Okay, and I'm telling you, it is not easy to start with something which is not there and then finding actually it is there. Yeah, and this was, I think, possible only because of Vijay. Now, Nagaraju and Tejas are the people in the start who taught me how to go about fly and how to get Tejas. Tejas is giving a lecture somewhere in Pune. It seems even when NCBS interview, when he came in, I think that's his signature. Yeah. This was, I think, in Pune where he was called to give an inspirational talk. Yeah. It's very inspirational. And Ajit was somebody I personally think, you never know what this guy is thinking, and will slightly push you towards, what's the correct way. He will never tell you overtly anything. I remember distinctly when I was given Comprimog, this guy, first of all, he doesn't talk in as loud, pitch as loud as me. He was sitting back in the row, and he would ask questions in minimal possible pitch. Now you ask this guy same question to repeat, he would do the same thing and just intimidate you, just to see whether I lose my patience or not. So, and then eventually he told me this is what he was doing, and he was successful in doing that. But still, Ajit remains as an inspiration. Ajit has done fantastic in his postdoc, as we all know. Now, Bhaskar has joined very recently, I think a year back or so. And since he has joined, I've learned a lot in terms of postdoc and how postdoc's life is. And he's, and I'm a bit scary to take that path at this moment now. Thanks to Bhaskar for that. And it's a good friend of mine, Guru, who actually, when I was generating data and things like those, was a very good critic. And still we exchange ideas and things like those. And I think these were the people who helped me, besides all of you in the lab. Yeah, but these people really, I took some time of them. Now, going back in the history when I joined, Das I forced to attend this presentation. Das was very patient with me when I was making this project for comprehensive. Now the comprehensive pattern has changed. It was different before, and Das helped me to design this project on planarian regeneration. And after designing all that, and when I was supposed to present, I didn't answer any question. But Das really helped me in that, and that's why I'm here. So thanks, Das. And Ram Kumar was somebody with whom I discussed my most of the muscle studies and hypotheses. Ram is now in Hampi, and I'm going to give the same presentation to him when he comes back. So yeah, he's not spec. And Raghu and Manisha, these were the best TCM members. I would say I troubled them a lot. And personally speaking, my workbench is right next to Raghu's cabin. And this is how I talk. So I think I must have disturbed him. I'm sorry for that. But this is what I'm inbuilt with. And Manisha also, she was very supportive, and in one of the conferences she actually told me to calm down and how to practice and all. So I thank Manisha for that. And she, I think, sent the very weird looking picture of mine. I thank for that also. And Girish and Talila took time to read my thesis and suggest me corrections, which has led to addition of 3 to 4 pages where we discuss how one can go about niche in general, because these were the same concerns raised by reviewers, and there is a lot to do in terms of niche signaling and all. So we think they were valid enough suggestions. I have done all the writing job, not done any experiment on that. Talila also was very nice, and she has suggested me some corrections in thesis, and they also helped me to really understand what was going in the study. Now, Du Jopan and one unknown reviewer were very helpful when we were communicating this paper to eLife, and they gave constructive comments, and this has helped to understand more on wingless pathway, and those results actually are resultant of these suggestions by these reviewers. Now, what I understand since I have joined this campus, there are many scientists who come regularly, and Ishwar Hariharan, Vivek Jayaraman, Benny, Helen, Malhotra, and Jyotsna. These are the great scientists, and I think I have personally learned from them how to go about science. And especially Ishwar, I remember when I was in second year, was somebody with whom I discussed first time. This was the first scientist with whom I was discussing my science. And he asked me, when I was showing this phosphorus along with GFP staining only phosphorus in the first layer, he asked me, do these cells pop out from epithelium or not? Now till that moment we were just scanning tissues and all, and that was the moment, a eureka moment for me at least. And then I went back and looked at the scan, and it was indeed the case. So a lot of credits to Ishwar. And I'm not naming friends from INSTEM and IISc, TIFR, JNC and NCBS because there are lots. And I like especially talking friends, so I have lots of friends. And NCBS kitchen staff, it's one remarkable group of people, never complain, though there are times when we don't remove labels from the vials and all, and they were very considerate all this time. And the amazing imaging facility we have, and I think the credit goes to Krishna, because he being here, we can see most of the microscopes working very well. And amazing people at reception, for especially Anand and Basavaraj. These are the people at your, to help you anytime of day, night. You call them, they're ready to help. And we have actually sometimes some people have misused this facility.